everybody. Thanks for being here. I know I'm the last thing standing between you and your lunch, so thanks for spending the time here listening to the presentation. So let me introduce you to a pretty different company than the ones you've heard before. We're not developing a therapeutic or a molecule. What we're developing is a technology to help pharmaceutical companies, being animal health companies or human health companies, get to market with a technology that allows them to produce proteins in a much more efficient way, scalable way, and simpler way. Okay, so who are we? This is us. We're a private held company based in Spain with a very solid scientific base. You have our founder sitting here, Jose Escribano. He's been a scientist for more than 30 years, pretty well known as a scientist. We have a solid number of partnerships with private and public centers, robust IP across disrupting technologies. So we have two technologies which are taking forward, TopPack and CRISPR. Both are patent protected with granted patents in Europe and the US, and production plant, laboratories, and headquarters in Madrid. This is the team taking the company forward. So this is the management team. Myself, I'm the general manager. I have a background in human therapeutics. I spent 13 years at Algenex doing the licensing for them, did the license deal with Takeda that then brought on the acquisition of Algenex by Takeda, which was concluded in July 2018, and following which I then joined Algenex as CEO. Jose, as I mentioned, is our founder and CSO. He founded the company in 2005. He was in the academia until October 2018, when he then joined the company full-time following our Series B. Virginia comes with me from Algenex, where she spent 13 years as a finance director. And Romy Dalton, she's our chief operating officer. She's been with the company for the last 5 years, taking care of all the operative side. We also have a pretty well-known strategic advisory board composed by people who are well-known both in animal health as well as in human health and in the investment community. So René Arts, he's a former member of the executive committee of MSD Animal Health. Jean Stéphane, he's the former member of GSK's executive committee. Claudia D'Agusta, she's brought IPO'd a couple of companies, including Algenex and then Theracon, which was then sold to Pfizer and is now in the spin-off of Theracon, which is Vectiv Bio. Damia Tormo, who's a general partner of Columbus Venture Partners. And Roberto Nicola are two gentlemen, Italian investors who raised the Series B, putting together a funding vehicle in which most of these people have invested. Okay, these gentlemen spend with us, and gentleman and lady Claudia, they spend with us a full day every 3 months supporting us and guiding us in the strategy. So what are we trying to do? As you all know, large molecule biologics, being proteins, they are the real center of drug innovation today. And they're used for a variety of things. They are used as therapeutic agents based on their role in cell biology. There are also structural elements of pathogens, and as such, they are part of vaccines. But they are also commonly used as reagents in vitro diagnostics. These are just 3 of the many applications that they have. They are also used as enzymes, hormones, etc., etc., etc. They all have a common thing, which is they're extremely complex to manufacture. So they are too complex to be synthesized chemically, meaning that they need to be manufactured in living, adequately programmed cells. What this means is that expensive and costly equipment is needed to maintain cell viability or productivity. This equipment is essentially bioreactor facilities. They're validated, which is a nice thing. They know they work, but they have a number of limitations. So in terms of complexity, scalability, versatility, yield, and cost. Okay. So what are we trying to do? We're trying to change the way the manufacturing is done and give a solution to these limitations. How? Well, we have seen, and it's widely documented, that insects have the capability to produce complex proteins. So what we're trying to achieve is change the way proteins are manufactured by, in essence, doing the same thing that the industry is doing, programming insect cells through a recombinant baculovirus vector, but instead of putting them into bioreactors and getting the purification and obtaining a final product, we're using living organisms. So we're infecting Trichoplusiani, which is an insect, which is the natural host of the commonly used baculovirus vector in the industry, that has millions of cells in perfect physiological conditions. And we're infecting this insect with the baculovirus that we develop in-house, and then we do the same thing, purification, and obtain the final product. Now you will ask yourself, so is this regulatorily approvable? It is, and there's actually a precedence on the market. There's a product that is commercialized by Virbac Pharmaceuticals today. It was approved by the EMA in 2004. So what's the difference between that product and us? Why do we claim that we have an innovation that it's worth financing? There's two main differences. The first is the choice of insect. So Virbac and Omega uses silkworms as production element. We're using the Trichoplusiani. So the Trichoplusiani, as I mentioned before, is the natural host of the baculovirus commonly used in the industry. That allows us to have much more productivity. But also the second innovation, which I think is even more important, is that we're using chrysalis as a production tool. So instead of using the larva, which has been used for the developments, we take the chrysalis, which allows us to automate the process and obtain scalability. Okay, just for you to understand how much we are talking about when we speak scalable, in a 9-week cycle, we're able to produce around 250 million insects in our facility. And we can obtain in each of these insects between 10 and 80 vaccine doses per insect. Now, if you make the numbers, you will see that there is a huge scalable system. So this is a small video that shows you how we rear the insects and how the whole thing is actually done. As I mentioned, one of the advantages, or their huge advantage, is that we have automated the process. And you will see here how we seed the eggs. We rear them in specially designed boxes. The larvae are grown through all the stages, and then they pupa in these boxes in around 2 weeks. And then we go to the automation. So the boxes that we have designed, they go into this machine that eliminates the silk from the pupa. It places them in a pick-and-place robot that we have designed ourselves. And then those trays go into an automated baculovirus vector inoculation robot. Okay? Each of those trays contains 160 pupa. And as I mentioned before, each of these pupa, depending on the antigen and depending on the dose required, can produce between 10 and 80 vaccine doses. Okay? So just to sum up about CRISBio, we have a new disruptive system that allows us to have enormous productivity of gram per liter levels, production flexibility thanks to the linear scalability, and this is very important for us. For us, it's the same cost producing 1 dose than 100 doses than 1 million doses, and we can scale up or scale down very fast. Okay, it's simple and robust thanks to automation, so the manual interactions are reduced, and we have an upstream cost per vaccine dose that is much less than 1 cent per dose. Okay, obviously, you then need to add the downstream cost to this, but compared with the cost that is right now in the industry, I think this is a substantial benefit. To talk about simplicity and cost, so this is just a small comparison between the currently used system, so the bioreactors in our system, and we're taking here as an example an animal health vaccine, which is PCV2. It's one of the best-selling vaccines today. To produce 50 million vaccine doses of this vaccine, you would need around 1.25 kilograms of protein. If you were to use bioreactors, which is how the vaccine is produced today, you would need to invest into several wave bioreactors, then small-scale bioreactors, and a larger-scale bioreactor. And the average upstream cost of an experienced baculovirus company would be between 5 and 10 cents per dose. But on top of this, you would have an estimated fermentation volume that would be above 8,000 liters. That's the fermentation volume which you have to take into the downstream. Okay. If you use CRISBio, you would need around 250 trays of pupa and a 350-liter incubator, which is like a fridge in your house. From this, you would get an infected pupa biomass, 8 kilograms of infected pupa biomass, containing around 37 grams of CAPD each. So you need a total of 282 kilograms of biomass, and you end up with 4,500 liters of extract, okay, in the same time. But you have several advantages. First, you have no capex. You have very limited OPEX. Again, we have less than 1 euro cent per dose per upstream cost. And above all, we have around half the volume to start with the downstream, okay? So this is, again, just one example of what we're producing today. The platform is set up to work for a number of proteins, as I mentioned before. We are taking forward a number of vaccines. We've started in the veterinary field, simply for the reason that it's an easier pathway to regulatory approval. So as I will speak in a minute, we have the first vaccine for an animal health indication under review today by the EMA. But we have also taken forward a project internally for avian influenza that we're developing as a proof of concept to enter into human. I do not have the data here, but just to tell you, we tested a construct in an animal study and saw 100% protection in the animals with the vaccine that we tested. And the next step will be to take this vaccine forward into ferret trials, just as a proof of concept to go then later on to humans, which can also be the entry point into a program for pandemic preparedness. Obviously, for the coronavirus that's actually running right now, we will be too late, but later on, this may be a platform that will allow us to enter into pandemic preparedness. Why? Because it takes us between 7 and 9 weeks to develop a new antigen, so we're a very, very rapid platform. We also have some assets in diagnostics. We have an existing partnership with BioKit in the human field, and we have another one with Global DX, a Scottish company, in animal health. And we also have a couple of programs with antibodies. We have historic experience with omalizumab, and we are right now taking a project forward to develop an antibody for an animal health indication. This is the initial focus that we have. As I mentioned, we started with animal health because it's a faster pathway to market regulatory-wise. So we have a vaccine for the prevention of bovine diarrhea in partnership with an Argentinian company. It's actually on the market in Argentina. We have a dual rabbit vaccine, a couple of vaccines in swine. Then the little chicken that you have there is actually the program that we took forward internally for avian influenza as a proof of concept also for humans. And the project to produce antibodies for the treatment of dogs is underway. 2 partnerships for the commercialization of human and vet vaccines. This is the press release that was issued when we submitted the first vaccine to the review by EMA. It's actually not ourselves who submitted it. Our business model is to work with partners. So how this works is that a pharma company, human or animal health company comes to us wanting to do a proof of concept to see if we can develop the protein, produce the protein in a more efficacious way than they're doing right now. And then we license that compound to them. They source it with us against milestones, royalties, and then we supply the protein going forward. Upcoming milestones, we have the CVMP opinion of the first Chris Bio-produced vaccine coming up end of this year, beginning of next year. We're moving into a new facility with capabilities to do GMP production, and we hope to bring the first proprietary diagnostic on the market sometime this year. This was all financed through a Series B that was raised in July 2018 with 2 closings. The last closing was done in September 2019. And we have 3 more milestones on our list, which is the expansion of the product portfolio, expanding into GMP, and doing the proof of concept in human health. And for this, we will need to raise a Series C sometime mid of this year. With this, I end. I don't know if you have any questions.